Actually, so before we start talking about, the two papers of yours that I want to talk about, the Deforming the Metric of Cognitive Maps and Navigating Cognition, I'm just curious, how did you find out about grid cells or place cells or whatever, and how did you end up getting interested in doing this kind of research?
That's very interesting. So I studied psychology as my undergrad, and I got fascinated by memory research mostly. And so I worked with Charan Ranganath at UC Davis for a few months, and then kind of was determined to do a PhD in memory research. And I found this position in Christian Jeller's lab that I signed up for or applied for, and was very lucky to get. And yeah, that's kind of how the whole thing started. And in the beginning, my first PhD project was on sort of these grid-like signals and imagination and memory recall. So that's sort of the link between my initial interest in memory and then fitting in the space or the navigation stuff.
But you'd heard of grid cells before you applied for the position, or?
Yeah. I mean, I'd heard a bit about them, but not so much. And
Okay. Was the first one, is that the eLife paper in Donderstown?
Yeah, exactly. Yeah.
Okay, so you really came through memory. I think that's kind of unusual, right? I feel like a lot of people who, when they hear about grid cells or place cells, at least for me, I heard about it in my master's for the first time, and I almost did a research project on this. And for me, it was just like this immediate, like, oh, this is really cool. I want to work on this. And
yeah, no, I definitely had this feeling of being fascinated by really learning about it, right? But like, I feel I only really learned about it during my PhD. Like before, I had some superficial knowledge, and my true fascination only started when I dug a little bit deeper.
Oh, cool. Okay, so as I mentioned before we started recording, we probably should at least very briefly mention a few keywords. So can you, in, I don't know, a minute or two, it doesn't have to be long, say what place cells, grid cells are, and maybe what a cognitive map is, I guess.
Yeah, maybe I'll start with the place cells. We can go in chronological order. So place cells were discovered by John O'Keefe in the early 70s. And what John O'Keefe did is he recorded the activity of individual neurons in the rat hippocampus. And he did that while an animal was moving through space. So the animal was moving through a little environment, a little box. And what John O'Keefe noticed was that specific cells were most active when the animal was located at a specific position in space. So for example, the northwest corner of the environment. And so one cell would always fire whenever the animal was at that location. And so he termed these cells place cells. And the idea is that each cell has a preferred location or where it exhibits a so-called firing field, and together the population of place cells could provide something like a map of the different locations that are known in the environment. And so if we think about what a cognitive map is, then it's maybe like a representation of different locations in space that is somehow true to their relationships. So for example, the distances and directions between positions in space can be read out from this map. And then maybe the third component or the third term you mentioned are grid cells, and these were discovered by Maybrit Moser and Edward Moser, and they were discovered in the entorhinal cortex, so one synapse upstream from the hippocampus. Unlike a place cell, which is active at one position in space, a grid cell is active at many locations in space. And these locations, they're not randomly spread throughout the entire environment, but they're distributed in a very regular pattern where the individual firing fields form the vertices of equilateral triangles that tile the entire space. So you end up with a super regular 60-degree symmetric firing pattern. And that's actually how they got their name.
Yeah, and so maybe if funny thing is, I think explaining place cells is pretty straightforward. Explaining grid cells gets very difficult without visual aid. So I think if this is completely new to any listener, just Google grid cell and you'll probably see an image when it becomes pretty clear what it is. But it's always funny, like when I asked, Matthias now did the same thing, and he also went this like, oh God, how do I explain grid cells in a way that's easy to understand just using words?
Yeah, definitely. It's very helpful if you look at this pattern. It's quite striking, but it's quite difficult to describe.
I mean, maybe we could say it's like a chessboard, but not with squares, but with hexagonal patterns.
Yeah, exactly. Or like a coordinate system. Maybe this is a metaphor that we can use a bit, right? So it's like a coordinate grid in a way, but they're not squares, but hexagons, basically.
Okay, and I mean, so these were all found. I mean, this all started in animals, right? But then your PhD supervisor, Christian Döller, with Neil Burgess and Caswell Barry, they found a way to do this in humans. And I guess this is what, I mean, you've only been working in humans, right, with fMRI?
Yeah, exactly. Yeah, I work with humans.
Yeah. And I guess that's kind of what we'll be talking about. I don't want to, I will get to the abstract stuff later. Mm-hmm. But I think maybe it might be interesting to mention it briefly that even though all of this was found for spatial navigation, in the last, I don't know, 5 to 10 years or something, that quite a few papers have come out of people showing that this entire system might be used for other dimensions or variables, basically. So it's not only the X and Y axis of space, but X and Y can be any axis. But yeah, we'll get to that later. I thought it would be just nice to mention it at least briefly in the beginning.
Yeah, and maybe one interesting thing to note there is that this is kind of coming back to Edward Tolman, who coined the term cognitive map, right? So in his work, which was based on navigating rodents, no recordings of neurons, but he discovered that they were using something that he referred to as cognitive map, and he already speculated that we might have these cognitive maps also for higher level psychological function. And I find it super fascinating that now, decades later, we're sort of circling back to these ideas.
Yeah, it's funny about this whole thing, right? When you feel like there's something that's super new, but then there's a guy in, what was it, 48 or something, writing about this.
Yeah. Some cognitive psychologist has done this 40 years ago, whenever you think of a clever paradigm.
Yeah, I mean, I've had that a lot when in my stuff. You think you have a cool idea, and then you find like some paper from the 70s or something. I mean, often one thing I've at least found is that they then weren't often done quite as well, which is maybe why they weren't, the findings aren't known as well, because of the way the experiment worked isn't quite as, the evidence maybe isn't quite as good or systematic as you'd like to have it. But I mean, with Tolman, it's fairly, I mean, that's well known, yeah. But actually, this is something I underlined in your science review. Did he actually coin the term cognitive map? I always wondered like where it started, or
I think he was the one who started this, yeah. But I'm not entirely sure now. But
okay, yeah.
I think he's the one who really started using this also for humans, right? I think the 48 paper is called something cognitive maps in mice and men and something.
Yeah, and I think I mean, it's been like 2 years or something since I read that paper, but I think it also gets kind of weird towards the end because it gets really political or something.
Yeah, it gets quite political, and it's also reflective of the ideas of how human psychology works at that time, right? It's quite different in terms of how they perceive or how he describes the human mind or human psychological function.
Yeah, but apart from that, he seems to have been pretty spot on with a lot of stuff. Anyway, so I guess I'll make a very rough transition here. So I want to talk about your paper, Deforming the metric of cognitive maps. Sorry, Deforming the metric of cognitive maps distorts memory. Maybe the easiest way is if you introduce it, just kind of like or maybe first, all these first experiments were usually done in square boxes or round boxes or whatever, right? So I'm just, maybe can you kind of introduce your paper that way by saying like how the shape of the environment in animal studies has been found to have an effect on this?
Yeah, so this is actually or the standard grid cell recording study that is done in rats or mice is that the animal forages for food. The experimenter throws some cookie crumbs into the environment, and the animal happily runs to pick them up. And this is typically done in square boxes that are fairly small, maybe 1 square meter or so. And that's where you get these typically, or these canonical grid cell firing patterns, right? And then I think in 2015, a couple of papers came out, one from John O'Keefe's group and one from the Moser lab, about the impact of environmental geometry or environmental boundaries on grid cell firing patterns, that sort of suggested that boundaries have an impact on grid cell firing, and that this can actually lead to distortions. And in particular, it's the study from John O'Keefe's group, which was led by Julia Krupich, and Caswell Berry was also part of it, where they recorded grid cells in a trapezoidal environment as well as in a square. And what they showed is that these typically regular patterns that you get in the square are distorted in the trapezoid. So they became less symmetric, and they became sort of expanded in the trapezoid relative to the square. And we were sort of wondering whether there might be any functional implications of these distortions.
And I haven't mentioned it yet, but I put the references for the papers we mentioned in the description. So you don't have to look for those. You can just, I guess, look in the description. Yeah, I mean, one thing that I kind of found funny about your paper is that I read it, and then I think it took me, like, I got halfway through the results until I realized there wasn't any fMRI. Somehow I was just so expecting an fMRI study because it was about like grid cells and cognitive maps. And somehow I was like, wait a minute, there isn't any fMRI. So was that so for you, the behavioral implication was the starting point, or how did it kind of get going?
Yeah, so there are actually two reasons why there's no fMRI. So one is we really wanted to look at behavior, because I think so everybody sort of assumes that grid cells are very important for navigation, and if we think about cognitive maps, then probably we think they are also important for memory somehow. But actually, this link is not as established as we would maybe like. So, for example, typical rodent recording studies, there's often not really a task, right? Yeah. So there's no memory task. And also so there's some evidence that these fMRI grid proxy measures might correlate across subjects with spatial memory performance, but within subjects there hasn't really been any investigation. So this is one reason why we really wanted to focus on behavior. And then the other aspect, while we actually couldn't record any brain data here, is that we wanted to use immersive virtual reality, where participants were actually using a head-mounted display, so like the Oculus Rift, and they were moving on a motion platform. So this, of course, prohibits recording fMRI at the same time. And the reason why we really wanted that is that we thought if there's an impact of boundaries on our spatial memory abilities, then probably it's rather subtle. And so we wanted to maximize that impact. So we wanted to be as immersive as we could be, and that's why we went for this behavioral experiment. And there are also some more technical issues with recording this hexadirectional grid proxy measure in environments where the sampling is biased, because it's an analysis that's based on evenly sampling movement directions through space. And so if you have a trapezoid, then your directional sampling will be biased. And this also it's potentially a solvable problem, but it causes some issues.
Yeah. I mean, it's not a criticism of the paper. It's just somehow, I guess, because I know the kind of studies that Christian Döhler does in his lab, and because I think like most of the papers in this area I've read have done fMRI. So it was more like just my priors were so strong that it took me like quite a lot of words to get that to start questioning that assumption. But yeah, so one thing I was curious about is that, so I mean, as far as I understand, in most of the standard human grid cell studies with fMRI, people use, it was kind of like a computer game, right? You press buttons and you move through this kind of arena or whatever.
Exactly, yeah.
Has anyone actually done a study where they compared whether, I guess, the technical limitation is pretty obvious here, but I'm just curious, do you think it's going to be like, does it make a difference whether people actually move through the space or just watch an avatar kind of move through the space? Because in a way, that's an assumption you have, right?
Yeah. So, I mean, I can only speculate here, but so from my personal experience using this VR setup, it's much easier to navigate because you simply have more cues, right? So you have cues about your body rotation. You kind of have proprioceptive cues about the number of steps that you take, and these make it a bit easier to navigate and to remember different positions. And I never formally tested it, but based on my experience with these desktop-based computer games, contrasting this to the immersive setup, I felt that participants were better in the immersive version in learning positions, like they were a bit faster to pick it up. And I guess it's just easier to keep track of your orientation. And then relatedly, in rodents, there's interesting work now that tries to use VR in rodents to sort of dissociate the contribution of different cues, like visual cues or self-motion cues, on the formation of grid cell firing patterns. And
you mean like because you can manipulate these in VR, but not as easily, at least?
Exactly, yeah. So if you use VR, you can disentangle them, right, by manipulating how fast you move for a given number of steps.
How do you know much about that? How does that? I mean, I saw a very funny image of like I think a fruit fly on like some sort of ball with a huge screen in front of it, so the fruit fly could kind of walk through this virtual space. Is it like that, or how do you do it with rodents? Do you know?
Yeah, I think it's a similar setup that you basically have a large, like a trackball that the animal runs on. So they had fixed often, or sometimes. So there are also groups that have setups where the animal can turn their head and move around a little bit. But in essence, the animal walks on like a styrofoam ball, and there's a big screen that surrounds the environment. Yeah, or surrounds the animal.
Yeah, I just love the idea of. I don't know. It's just inherently funny to me, the idea of these rats being in some sort of VR.
Yeah, and doing VR and fruit flies. I mean, that's just amazing.
Yeah. Okay, so it's something that kind of seems, it seems like it might be more, but you didn't feel your grid cells firing more or less in one or the other.
I think some variant of this question is to be asked to Matthias Stangl, right? Because he records signals in freely moving humans.
Oh, that's a good point.
So yes. Maybe soon we'll know the answer.
I'll have to ask him that, yeah. Oh, yeah, because he doesn't have the fMRI limitation, right?
Exactly, yeah.
Uh-huh. Actually, I wrote that down because it never occurred to me that, this guy I'll be talking to soon might actually be the person to ask this question. Anyway, so how did, I guess the question is kind of like how did you decide to do this study? Was it you read this paper by Krupic et al. And thought, let's do something with this? Or how did it kind of, or was there already like this question before where you thought like, hey, I wonder whether grid cells actually...
Yeah, so this was definitely something that I was interested in before. So how do grid cells relate to cognitive function, right? So how could they relate to memory or imagination, for example? That's something that I worked on before. And so I was definitely interested in that. And then the idea was sort of that we could maybe use these distortions as a window to look into how this relationship might come about, right? Of course, whenever I say this in today's conversations, it's with the limitation that this is a behavioral experiment, right? And we didn't actually record any brain signals.
But I thought it was a very neat way of, I guess, how should we say, sidestepping the technical limitation of not being able to record fMRI in this context. Yeah, maybe, I guess we've only introduced the paper. Do you want to say briefly then what you found?
Maybe. Yeah, I think I haven't even really explained how or what we did, right? So in our experiment, participants navigated two environments. So one was a trapezoid and one was a square, and we'd matched them for a surface area. And in each of the two environments, they learned the positions of 6 objects, and then we probed their spatial memory. So we asked them, we showed them a cue with an object image, and then the task was to navigate to the position in the environment where they think the object belonged. And as I said, we used this highly immersive VR setup. And we also test them afterwards in a simple desktop-based experiment. And so for the first part, what we were interested in was we wanted to look at how precisely they could recall the positions of the individual objects, right? So basically, we measured the distance between where they think the object belongs and where the object actually belongs. And what we saw then was that participants' memory for the object positions was less precise in the trapezoid than in the square. And we also found evidence for a more fine-grained prediction that we had, namely that within the trapezoid, memory was particularly bad towards the narrow ends, towards the pointy part of the trapezoid. And this is exactly what Julia Kupich had reported about the grid cell firing patterns in rats.
One thing that really surprised me reading this, I mean, I haven't read the Kupich paper yet, so I wasn't aware of them having found that. And it kind of surprised me when I read it in your paper, because it seemed kind of counterintuitive to me, because I would have somehow imagined if you're closer to borders, it would be easier to remember this. But yeah, obviously that's not the case. So
yeah, so this is definitely an expectation. And it's actually something that we confirmed that this is the case in the square. There it seems to work like that. But in the trapezoid, somehow this seems to break down. And so it's something that should work against us, right? So it should be easier to do this in the narrow end, because you're closer to the boundaries. You should actually have more cues about where you are. But somehow this effect could only be seen in the square.
Do you have any idea why?
Well, I like to hope. Yeah. I hope to think that it's somehow related to the grid cells. Yeah. So I mean, typically, I think we use boundaries to kind of anchor our cognitive maps, right? So to anchor our representations of space, we maybe use the corners of a room, and I don't think so, or quite obviously, our participants weren't lost in the environment, right? It's still a fairly simple environment, but precisely locating where something belonged was more difficult in the trapezoid than in the square, and more so in the narrow end.
I mean, is it maybe because, I don't know, in the square you have both the, so let's assume it works exactly the way it does in rats and humans. So in the square you have both the boundary vector cells and the grid cells, whereas in the trapezoid you kind of only have the boundary vector cells because the grid cells kind of break down? Or
yeah, I mean, they don't break down entirely, right? So the regularity of the pattern breaks down. So your basically your memory code might be just less precise.
Yeah, of course. Yeah, it's not like they just have no idea where they are. Yeah. I think if that was the case, we would have noticed already.
Yeah.
Just in everyday life, like as soon as we're not in a square room, we just have no idea where we are.
Exactly. Yeah.
But it is kind of surprising, though, right? That just as I said that, it did strike me like this should be something. I don't know. It feels like if the shape of the room you're in has such an effect on being able to memorize things, that we would kind of know that. But outside of these basically two studies, I've never heard of the idea that.
Yeah.
I mean, or is it just like a fairly small effect that? Yeah, I don't know. Yeah,
I mean, so it is a study. It's a lab study, right? So the situation is kind of artificial, and generally people were very accurate in remembering these positions, right? So they knew very well what they were doing. Maybe anecdotally, the building at the Donders Institute where I did my PhD, that's a triangle, and that's quite confusing in the beginning when you walk around the corridors and you end up in the same place basically. Sooner than you thought.
I have never been in a triangular building, I don't think.
Yeah, but I mean, it's a fair point, right? We're much more familiar with rectangular rooms or buildings.
Do you think? I'm wondering, are rats more familiar with square rectangular rooms than with trapezoids? I mean, I guess the ones that grow up in the lab probably are, right?
I guess they have like the home cages are typically rectangular. Yeah.
Yeah. Who knows? Maybe it's just a.
But so one interesting thing about the Kupich paper is that this distortion was very stable over time. So I think they recorded definitely over multiple days. I want to say even 2 weeks, but
yeah, okay. So you'd figure if it was like a learning thing, then
if it was just simple. So they are known to be some like transient effect of the expansion of grid cell firing patterns when you put them into a new environment. And then it's a sort of novelty effect. This is something that Caswell-Berry, for example, has shown. And so it's not that, but it's something that seems to persist over time.
Okay, cool. Yeah. One question I wrote down that I think you've kind of already answered in part, but I'll still ask it, is that, I mean, one thing I find kind of interesting about a lot of Christian Dollar studies is that it seems a lot of it is a fairly straightforward translation from findings that have been done in animals to see whether we can test it in humans also. My question was kind of whether your study is a straightforward translation in terms of like, is this possible or not? Or whether there's also kind of an extension of it that kind of shows something that we didn't know before. Yeah, I think maybe it seems like the memory aspect might be.
Yeah, so to me the new so had we basically done an fMRI experiment and shown that the hexadirectional signal is weaker in the trapezoid than in the square, I would say this is like translating that finding. But because our focus here really was the behavior and memory, I think it's I think we're kind of trying to use the finding in rodents to learn something about human cognition. And this is actually one aspect that I really, really liked about working on this project, was that basically we're taking this finding from systems neuroscience and we're trying to figure out if this maybe has a relationship to human memory, right? So this kind of high level human cognitive function.
Yeah, and I'm assuming the Kuper paper didn't have this kind of memory aspect to it.
No, I think this was a free foraging task.
Mm-hmm. Okay, yeah. So I'd kind of like to leave the wait, what's the word?
Trapezoid?
Deformed. Yeah, or like the deformed spaces kind of on pause for a second, because kind of I want to talk about this in the abstract cognition sense. So I'll just pause on that for a second and come back to it later. Before we go to the kind of more abstract cognition thing, one thing I've always been curious about is that, has anyone done a grid cell study in non-Euclidean space? Let's say you have like a rat moving around on a globe or sphere or something, like, because the kind of basic question is, in a lab we often have, it's always a flat arena, right? But obviously, if you walk up a mountain or something like that, then it's very different. I mean, I've talked to Kate Jeffrey on the podcast about her work in 3D, where you have the rats really climbing up and down. But I'm, just as a kind of curiosity, I'm not aware of anyone who's looked at if basically you have a surface that's not flat in this sense, right?
Yeah, I'm not sure if anybody has done that in rodents, not that I know of, at least. And I guess it's kind of hard to do, right? Yeah, walking on a sphere is quite abstract. There's some work going on in our group. Actually, there's a recent preprint by Mi-Soon Kim, who's a postdoc in our group.
Okay. And
she looked at this. She used this basic object position memory task on a curved surface, so kind of similar to what you outlined.
Yeah, that sounds pretty similar.
This is behavioral work as well, so you should definitely check that out. I'm not going to spoil it.
Okay, yeah, and I'll if I can find it, which I can, I'll put a link in the description. Okay, cool. So abstract spaces. I mean, this is the thing that I found the spatial navigation stuff really interesting and fascinating, and it's really one of the things that kind of got me really interested in neuroscience. But in a way, it's also kind of not that interesting because it's just about moving around. And the thing that's kind of most interesting, to me at least, is this set of papers that came out in the last few years that show that this is not just about moving or memory of moving or where you are in space, but that this applies, well, that's the question, right? I guess you in your paper, you say it applies to, you don't exactly say to everything, but you kind of say it's a kind of domain general thing that can, in principle, I guess at least, code for any variables. So can you briefly summarize the kind of the gist of your review paper?
So in this paper we kind of tried to bridge some gaps, right? So we worked together with Edward Moser from the physiology side and Peter Gerdenfors, who's a philosopher of mind, to sort of try to apply what we'd known about forming cognitive maps of space, and then these super exciting studies that you referred to that sort of came out in the past couple of years that sort of suggest that these spatially tuned cells like place cells or grid cells, also seem to map more abstract dimensions. And we, in our paper, we refer to these abstract spaces as cognitive spaces. And we tried to summarize the findings that had come out from different species about how the hippocampus and entorhinal cortex might build cognitive maps in tasks of different domains, and then we tried to outline how this would be important for high-level cognition. So, for example, for concept learning or inference and generalization.
Yeah, I think it's a really nice summary of there's this kind of whole idea, because I was aware of a few papers, like the Constantinescu paper or one or two others, but I didn't realize just how much had been done in this field already. And yeah, maybe you mentioned that you kind of combined the human work, the electrophysiology in animals, and the philosophy of mind work. And I have to admit, I'd never heard of Peter Gerdenfors. I don't know.
Mm-hmm.
I don't know.
Yeah, he's Swedish. Yeah.
Yeah. Can you? The book sounds super interesting and relevant, but I haven't read it. What kind of book is it? Like, and what kind of work does he do? Because I'm just not familiar with his work.
Yeah. So Peter Gerdenfors is a professor in Lund, and so he works on philosophy of mind, and he has this book, or his theory actually, on conceptual spaces. It's not neuroscience, so this is really philosophy of mind. It's his key work there. There is also experimental work a bit in that informs this, of course, but it's not really about the brain. And at some point, Christian Deller and I, or Christian, became aware of this and kind of pointed me towards it, and I read it, and I was kind of fascinated that some of these ideas seem to be very much in line with what we were seeing in the hippocampus and entorhinal cortex. So the general idea of this conceptual space that Peter Gerdenfors put forward or proposes is that space is a great representational format to organize knowledge in, right? So you can project knowledge into a two-dimensional space, and then you can define concepts as a region in this space. So one example that we could maybe use is, say we have different animals, right? So we can arrange them based on how they look, like how many legs they have, how heavy they are, and so let's say we have.
Those are all the categories we have.
That's all. That's everything we know. How fast they can run, whatever. How large their teeth are. That's probably relevant. And so let's say you learn that there's this concept of big cats, right? So these will be animals like lions and tigers and maybe a cheetah, and they will occupy a similar region in this conceptual space, right? So they'll have similar features, and so they'll be located close by. Now, one important thing is that if you organize your knowledge about these animals, or if you use a spatial representational format, then you can use very naturally similarity between these entities to generalize, right? So say you've never seen a tiger, but you know a cheetah and you know a lion, and you know that you shouldn't come too close because they're pretty dangerous to you. Then maybe because the tiger is similar, you can generalize and also not come too close, even if you haven't actually encountered one before. And so that's one way in which this spatial representational format is very powerful.
So a kind of, I mean, in some way, it's just reasoning by analogy and using, whatever variables are most obvious or distinctive or whatever about.
Yeah, I mean, this is then an interesting question, right? What are the dimensions that we use to actually organize something? And in the typical lab experiment, these dimensions are, of course, experimentally defined. So they're sort of, the experimenter decides what the relevant dimensions are, right? So if you, for example, think about the paper from Tim Barron's group by Alexandra Constantinescu, they have a space of different birds, and the birds, they differ in how long their neck is and how long their legs are. And so these are the two dimensions that sort of span the space. I think it's very interesting to think about more naturalistic settings, right? So or spontaneous grouping of stimuli that are not explicitly cued to differ only along 2 dimensions, right? But along multiple dimensions. And it could be that these are different depending on the context, right?
Yeah.
So in one context, it might be more relevant to know whether the tiger and the lion are similar in that they will eat me, and in a different one, it might be more important to, for example, consider where they live, in which part of the world you can actually find them.
Yeah, and I mean, this is kind of where my kind of own interests come fairly close to this, because what I'm kind of interested in is, I should say, like if you have a social situation where you have to make some decision that affects you and someone else or multiple people, whatever, you always have this like objective outcome that's going to happen, but people don't make the decision based on this objective outcome, but rather they have their personal preferences based on whatever context there is, who the other person is, what your history with them is, whatever else is going on in your life or in the situation. And so I'm kind of really interested in how exactly this translation happens, basically how these preferences affect it, and specifically how the brain does that, kind of how you go from this objective outside world to this kind of decision via these internal preferences. And one thing that I'm then really curious about is, for example, like let's say you have these 2 dimensions, and now they're not space or neck length and leg length, but they are, your payoff and the other person's payoff. Mm-hmm. For example, like how much money do I make if I choose X or Y? How much does the other person make? So I guess if you don't mind, shall we speculate a bit on how abstract cognitive maps relate to this? Mm-hmm. Or specifically, I'll just ask you to speculate more. So I guess the first thing is, so let's say I have one axis is my own payoff, the other is the other person's payoff. And depending on who I'm talking to, there's different, I like the other person more or less, right?
Uh-huh.
Do you think that would transform the actual kind of map itself? Or would you have this kind of objective map and then you would use that information afterwards at a later stage to, it's kind of like, in a way, like if we want to take the grid cell analogy, like would the grid cell pattern be distorted if I like myself more than you or whatever? Or would the grid cell firing still be stable, and then at a later stage we would use that information?
So just so I understood the setup, so you would have 2 axes of space. It's sort of this, is it called, the dictator game? That's one. I mean, there's lots of different games that can manipulate in various ways who gets how much and what the options are. Yeah. And so your idea is that depending on how much you like the other person, that axis is weighted more or less, basically.
Well, so let's just say we have 2 options, right? Either like I give, I should have thought of a good example. So let's say you have just a few different options. Kind of, you have, okay, let's just be specific. So there's the prisoner's dilemma is the most famous example. We have 4 outcomes, mm-hmm. and each of those outcomes gives you some money and me some money. And I mean, we both have to make a decision, but let's just think about like which one I want, which one's my preferred option. So I have 4 different outcomes with monetary rewards for me and you. And so these 4 points kind of lie on this 2-dimensional space. And I guess the question is kind of like, if I'm playing with someone I really like versus with someone I don't like at all, would kind of the space still be orthogonal? Would the grid cell firing still be regular? Or would this kind of difference in how much I like the other person just really like shift and deform the space?
Yeah, that's a super interesting question.
By the way, I'll just make this like, you can take as much time as you want, and I edit it down to like 1 second. So it looks as if you just immediately came up with the answer.
I think one idea or one speculation could be that the sort of map in the entorhinal cortex, if you like know all of these different options, could remain sort of normal, if you will, or undistorted, and that then the distortion happens somewhere else.
That would kind of probably be my guess, but I don't know the spatial navigation literature that well, and I wonder whether there's any equivalent study vaguely, you know.
Yeah, well, so at least there's some work in rodents that basically the grid pattern is also influenced by locations that are frequently rewarded. So if you introduce some regularities in the task, actually, then it can happen that the grid pattern already deviates from its canonical shape.
Do you know what paper that is?
This is work by Lisa Giacomo, I want to say.
Oh, okay. She has an interesting looking like review perspective article out that I want.
Yeah, exactly. She also has a recent Nature Reviews Neuroscience, I think it is, right? I haven't had time to read it yet, I have to admit. And actually, what you sort of described now is that basically there are 2 dimensions, and or what you described reminds me of a recent paper that came out from the group of Iri Burman in Nature Neuroscience, where they basically have 2 social hierarchies that form the dimensions of a space, and the task of the participant is, or the cover story is that the participant is an entrepreneur, and they have to select the one that sort of matches or maximizes their potential. And actually, they report some modulation of the hexadirectional code in fMRI based on this decision value, if I recall correctly.
Mm-hmm. Yeah, I still have to read that paper completely. I've only like skimmed it. Yeah, I guess maybe my question was a bit too in depth immediately. So maybe we can like backtrack a little bit and ask kind of. Yeah, one question I'm generally interested in is kind of like how far the findings from spatial navigation translate to domain general findings, or whether there are things that are specific to spatial navigation that just don't apply to other things. And one like maybe fairly straightforward question, doesn't mean the answer is straightforward, but the one question might be like, for example, what is a landmark non-physical space?
Yeah, exactly. So I think there are some challenges to these ideas, right? So what is a landmark? You could say a particular salient object that you know very well is something like a landmark, right? Or something like the prototype of a given category could be something like a reference point that you use, right? And there's plenty of cognitive psychology work showing that sort of this processing of reference points also in abstract concepts kind of biases how we process information. I think another limitation that's sort of related, that boundaries that are really essential to cognitive maps of physical space are much harder to come up with in an abstract space, right? Because I think what makes a boundary so influential in physical space is that you can see it. You can see it from afar. You can use it as a reference points. It gives you distance information, and these are things that are not easily implemented in these sort of abstract spaces. I would say it's much harder. And if you follow up on this notion that a concept is a sort of region in an abstract space, then there are boundaries between these concepts, right? So at some point you go from being in concept A towards being in concept B, but maybe in a real world naturalistic setting, these boundaries are not that sharp, right? They might be sort of fuzzy, making them much less suitable to serve as sort of reference points or anchors for your cognitive map, right?
Yeah, like whilst you were saying that, I mean, I was thinking kind of like what actually makes a landmark in these navigational studies. I mean, one thing is that they don't change, right? They're kind of fixed points that if you move, you can always evaluate your movement relative to this.
Yeah, so there's some work on that by I think by Eleanor McGuire's group, showing basically that one defining feature of a landmark is that it shouldn't move necessarily, right? And typically we use things as landmarks that don't move, right? So I usually don't use the car that's parked on the sidewalk as a landmark because probably when I visit this location next time, it's not going to be there, right? Much rather I'll use the bus stop because the bus stop will probably be there. But so in terms of how this position relative to a landmark is coded, there's some recent work in the physiology world about object vector cells, right? That basically give you your position relative to a landmark. These are cells that fire at a given distance and direction to a landmark that's in the environment. And then if you move that landmark, the firing field of that cell will also shift by a corresponding amount.
Yeah. But that's all in animals still, right?
This is in animals, yeah. But I think it's a great question to also try to tackle this in the human brain, and maybe in also abstract sort of dimensions.
Like one example I'm thinking about right now is maybe let's say something like one concept might be a person's height, for example, because it's still kind of physical. But let's maybe use that as an example. I mean, I guess could landmarks be something like, I don't know, for example, like 2 meters tall or something as like a kind of, or boundary or something like a cutoff point or something where you, or something that's like, I don't know, like I'm trying to think of something that really.
So I would have speculated that if you think about height, that maybe the height of someone that you're very familiar is something like a landmark.
Yeah.
So maybe the height of your partner, you sort of, it's easy for you to say someone's taller or shorter than that.
Sorry, I just thought of the perfect study. So you're going to use your parents as landmarks, and as you grow up, you move close to the boundary or whatever. So now all you have to do is just track lots of children and teenagers longitudinally.
That's brilliant, yeah.
Yeah, it's an interesting idea that's completely unfeasible to do. But something like that, right? I mean, it seems like you, or let's say, I don't know why I'm thinking of this example, but like when you watch a film or something and you're supposed to rate how good it is, maybe you have like your 3 favorite films or something that pretty much your appreciation of them is maybe unchanged or something. So then you view them as kind of like exemplars of what makes a good film or something like that. Yeah.
Exactly. Yeah. That you might have your favorite comedy and your favorite action movie, and you compare based on these reference points, basically, right? That you could use.
Yeah, but it's kind of funny to me how, in a way, it seems it should be straightforward, right? You have like, oh yeah, you walk through 2 dimensions, you've got, but then as soon as I don't know, it's something that to me at least seems like the application should be very straightforward, but it's actually much trickier. Maybe it's just because I don't work on the application, so it just seems easy from the outside because I only see the final papers. But I mean, no, I'm not saying that. You know what I mean, right? Like I'm not saying this work is easy. I'm still surprised, like how difficult I find it to think of the concepts that I'm interested in and kind of fit it into this framework.
Yeah. Yeah. So actually, to me, one of the things that I really like about this is that it sort of merges this very detailed knowledge that we have about how the system works in physical space. Like we know a lot, we learned so much from these studies in rodents and also from the navigation work in humans. Only that actually gives us the possibility to think about it in this level of detail. Like I think that's quite unique. That somewhere pretty deep in the brain, right, you get this sort of more or less abstract code for positions in space, right? That is sort of abstracted from other perceptual information, and I feel there's been amazing work on teasing apart how these representations come about. I mean, there's still so much we don't understand, but we actually do know quite a lot, and or we do know some very cool things about it, and these only allow us to ask these questions about how it might work for other domains, right? And that's why I find it so interesting to think about whether these mechanisms apply outside of the navigation world.
I mean, it's almost like a way to jumpstart creativity and having ideas about what you're thinking about, because you can just go like, okay, I'm doing this study. What's a landmark? Or like in this research area, what's that? And then you just have lots of new ideas.
And probably that's taking it or making it way too simple, right? So probably it's not just gonna
just for like idea generation. Even,
exactly.
right?
Yeah, you can brainstorm a lot, right? And you have cool ideas about what you can test. Yeah.
Yeah, I mean, it's funny, like whilst you were talking about the Coolidge paper, I was thinking this, we know so much about the navigational system, and yet you can, again, I'm oversimplifying completely, you can have a Nature paper in 2015 showing like, what happens if the box not square? Again,
so this was not their only finding.
Of course. But you know what I mean, right? Like it's I find it funny how often in lots of other research areas also, right, you have these like very these findings that are really cool, super well known and everything, and then you ask like some basic thing and it suddenly changes like how you view the whole thing. Anyway, before we kind of finish, I want to ask, I guess it's a fairly big question and something that you also address in this, which is how many dimensions can you do with this?
Right. Yeah.
Because that to me seems like a pretty important question.
I agree. It's a super important question. One thing that so there are different aspects to it, right? So on the one hand is if we're really coming from or trying to take the physical space analogy far, then we're going to get in trouble after 3 dimensions, right? And there's this super interesting work by Kate Jeffrey, who you had on the show, and by Nachum Ulanovsky, who studies flying bats, right, about place cells and grid cells in 3D spaces, and they do seem to exist. They seem to, at least for the grid cells, they seem to be behaving slightly differently from what we might have expected.
Yeah.
But I mean, things will break down if we go into higher dimensions. And if you look at human cognition or human spatial cognition, already then the 3rd dimension is often encoded less precisely than basically the flat surface, which might be because it's harder for us to we less frequently navigate in the 3rd dimension, right?
To me, humans kind of are 2-dimensional animals, right? I mean, sure, we can move like within, if we raise our arm or whatever, we can go up a ladder or something, but
and we walk through buildings and we've got that have stairs and so on, right? But
that's pretty.
We're not that great at 3D, and I don't know. Maybe we should test navigation in divers, something like that, to get like volumetric.
You want to do fMRI study underwater?
Yeah, exactly. Yeah.
Good luck.
Someone might drown.
I guess you just need an epileptic patient with the electrodes implanted.
Waterproof.
Yeah, exactly. I mean, so it seems to me there's kind of 2 kind of obvious ways in which this could go. Either it could actually be something that scales for however many dimensions you might want in a given situation, which potentially many, but let's be honest, usually is only a few that are really relevant. But so that's kind of one way, right, where it can actually contain lots of different dimensions at the same time. Or this is the other option that seems kind of obvious to me, is that it actually is limited to 3 or 4 dimensions, and the brain has to kind of figure out beforehand which dimension it feeds into it to deal with the situation. Do you have any speculation again about which one might be?
So one speculation for me is that we can break down these sort of knowledge kind of problems to dimensions that we think are relevant at a given time, and probably we cannot handle too many of them at the same time. I do think, and I mean, there's lots of work on the use of heuristics, for example, right? So we're not perfect at information processing. Actually, we're quite far from it. I think that that's possible. Maybe one thing is that there are also other accounts of how the hippocampus works that focus sort of more on transitions between different states, where defining individual dimensions is maybe not as central as to this idea of conceptual spaces, right? Where you really have to define dimension, or where you define dimensions based on what the stimulus features are. So these could sort of, or maybe they could yield testable predictions in cases where you go beyond something that you can represent with a 3D Euclidean map.
Yeah, so much to do. It's funny, like, when you first hear about this in my case, in my master's about spatial navigation stuff, you're like, wow, they figured this out.
Yeah,
like
we know this now, right? This is
exactly.
done.
And then once you actually do research, you go like, oh, this is just the beginning.
Yeah.
This is where it starts. But so do you think it would? It seems to me like your review paper is kind of, I mean, you address the question that this is an important question, but you kind of outline it, how should we say, independent of how many dimensions this can kind of encode for. Just showing like the general principle, and whether it works for 3 or 10 is maybe another question. But I'm just curious, how would your, I'm assuming you have also like maybe some slightly bigger theories of like how this all fits together. How would this change depending on whether the system can only do 3 dimensions or whether it can do 10 dimensions? Does that, kind of how would those 2 options, let's say, change how you think about how this fits into the bigger picture of, I don't know, memory, decision making?
I haven't thought about it from this direct angle. So, so to me, then, if we figure out that we can only do a certain number of dimensions, then I think the next obvious question is, how do we select the important ones, right? Because that's not always immediately clear. There might be situations where I ask you, I don't know, how friendly is this person? Then it's maybe clear. But if you just ask me, what about X? Then I can judge them on very many different dimensions. And so to me, this would, I think in both cases, it becomes interesting, right? How can we keep online so many dimensions and combine them? And do we maybe weight them equally? Or if we have to select them, how do we pick? Or do we sort of do some implicit dimensionality reduction, so we end up with some approximation of a higher dimensional space, right?
Yeah, and that there's so many links, but I think I think we'll have to stop it here, otherwise we're gonna be here forever. Because I mean, I find this really fascinating, and for me, I was really interested in the spatial navigation literature. Then, basically for fairly random reasons, I didn't end up doing research on it and ended up doing other things. And now it's kind of coming back, and I'm trying to kind of combine the 2. And it's just, as I said, it's earlier, like, I think it's a really cool way also just to have new ideas about what you're doing. Like, not you specifically, but about what one is doing.
Yeah.
And what one might do as an experiment. Maybe as a last kind of question, what do you plan on doing next, or what's your kind of current research? What kind of direction, at least?
Yeah. So at the moment, I'm kind of working more on the representations of relations that occur in episodic sequences. So the idea is that our memories kind of consist of different sequences of events. So my memory of our podcast recording will be doing this technical setup. Then we did a little introductory check, and now we talk, and maybe afterwards I'll have some dinner. And so I'm interested in how we represent the temporal relationships between these events, similarly to positioning them along a dimension in an abstract space. And so this is something that I'm currently working on, and looking at basically what happens if we have multiple sequences that have a similar structure, to look at whether the hippocampus sort of generalizes across sequences that are sort of similar. So I might have similar memory if I were invited to a different podcast. And basically, I'm looking at whether what we know from one sequence affects how we represent the other one.
Right.
Yeah.
Okay.
So this is it's connected, but it's a bit more memory focused. I would say.
Yeah, it seems like you're going back to back to
to
your
the
initial
roots.
interest. Exactly. Okay, cool. I guess this is gonna maybe take a while until that paper's gonna come out, but I'll read it.
Yeah.
Cool.